Hi, everyone. Welcome to the Physionic podcast, or welcome back to the Physionic podcast. If you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine. And, I guess that's basically all the credentials I'm going to be offering. I work with mitochondria, so that's going to be quite a bit of focus on this particular podcast. So part of my research is related to mitochondria. Now, last podcast episode, I went into some pretty severe detail when it came to breaking down a study that aimed to look at how exercise affects our metabolism as well as blood sugar levels. And we went into that in great detail, and I certainly encourage that you check that out if you haven't, mainly because that creates kind of a good baseline for what we're going to be covering in this podcast. But a lot of the content in this podcast is still going to be independent, meaning that you don't absolutely have to know everything from the previous podcast to follow along in this one. But that said, what is this podcast going to be about? So it's going to be the second half of that paper, second half of this particular study, where they wanted to investigate the amount of exercise. So from light exercise, moderate exercise, to excessive exercise, and then the recovery, and how that would affect mitochondrial dynamics. What does that mean? Mitochondrial dynamics is essentially how mitochondria react to a stimulus. So the stimulus in this situation is excessive exercise. So that's what we're interested in. And so we're going to look at two things. We're going to look at how the actual physical shape of mitochondria change in response to exercise. And we're also going to talk about how mitochondria are affected in terms of their health, in terms of reactive oxygen species, the damage that occurs to the mitochondrion, and as well as some of the components within cells as well. So they did a variety of different measures, these researchers did. And then I'm going to also offer a bit of information on a particular pathway, a signaling pathway, what might be causing some of the results that we're getting. All right, so to get a bit of background again on this study, just a bit of background on the actual mitochondria and a bit on this mechanism, as well as how exercise can potentially be harmful. And then I'm going to go into the study design, and then I'm going to go over the data. Now, if you're watching the podcast, of course, I'm going to have some of that data pop up, as well as some diagrams that I've created to make things easier. But as usual, if you're just listening to the podcast, have no fear. I will be discussing things in detail, describing them for you, so you won't have to hop on over. But with that said, so mitochondria, as people likely know, is a major source of cellular energy, right? People consider it the powerhouse of the cell. But in that production of energy, actually, it produces what are called reactive oxygen species. And these reactive oxygen species are a particular type of free radical. So I realize that's a bunch of random words, potentially. The idea is that these molecules that are produced by the mitochondria when it creates energy can do damage to the internal components of the mitochondria, but also on the components of the cell itself. So having an overabundance of what are called reactive oxygen species can lead to damage of the cells, because these reactive oxygens, they are oxygens that are, for lack of better words, unhappy, unbalanced, and they start ripping away subatomic parts of other molecules. So you have a functional molecule, those all make up the cell and the mitochondrion, and these reactive oxygen species are created, and they're unstable, and they just reach out essentially and rip away parts of functional proteins and fats and whatever components are part of that mitochondrion or the cell. So is it a good thing to have? That's actually a nuanced discussion. The answer is a little bit yes, because it's actually also used as a signaling molecule. So it tells the cell can communicate within itself using reactive oxygen species. However, if there's an overabundance, it can lead to damage of the mitochondria and the cell itself, which can lead to cell death. But it can also lead to mitochondrial reductions in function, and we discussed that actually in the last podcast. So again, I encourage that you check that out. And the researchers point out that reduced mitochondrial function is associated with increased insulin resistance. So talking about diabetes here, talking about blood sugar levels, things like that. So the ability for the cells to respond to the molecule insulin and to be able to drive blood sugar into the cells, so out of the blood into the cells. So the association between mitochondrial dysfunction and insulin resistance, and the link is attributed not only to mitochondrial number, I mentioned this again in the last podcast, but also to the respiratory capacity and the overall morphology. Now, we discussed, and what that means is that not only the number of mitochondria within a single cell, and not only their ability to produce energy, the respiratory capacity, but also the shape of the mitochondrion. And that's actually what we're going to be focused on here. We're interested in the morphology, so the look of the mitochondrion. So one of the regulations of mitochondrial health is through a pathway called NRF2, or NRF in general. And that's a truncated an acronym for the long version of the name, which is nuclear factor E2 P45 related factor 2. NRF2, that's what we're going with. And its relationship, that particular protein, that particular molecule within the cell, how that interacts with another protein, another molecule known as KIAP. So KIAP is a shortened version of Kelch-like ECH-associated protein 1. So the way it works, and I'm going to have a schematic that's going to pop up later on in the podcast if you're watching, but the way it works is that KIAP regulates NRF2. So NRF2 being responsible for the production of antioxidants. Antioxidants are molecules and proteins that the cell can produce that will combat these reactive oxygen species. They have the capacity to neutralize reactive oxygen species. So they're kind of the antithesis of reactive oxygen species. So the idea is that NRF2, if it can enter the cell nucleus, so the nucleus of the cell, where all of our genes are kept, where all of our blueprints are kept for all the proteins in our body, then it can bind particular genes that are responsible for the production of more antioxidants. Therefore, you have more control over those reactive oxygen species. But not only that, it also is implicated, NRF2 is implicated in its function in mitochondrial biogenesis, meaning the production of more mitochondria. So if you have more NRF2 around, and especially if it localizes, if it moves into the nucleus of the cell, then you typically have increases in more mitochondria and more antioxidants. So previous reports have shown that increases in our NRF2 are pretty immediate after exercise. So since exercise increases mitochondrial number, especially long distance exercise or steady state exercise, the researchers decided to look at this particular pathway. So that's what we're going to be looking into at the tail end of the podcast. However, of course, excess exercise, so going overboard on exercise, can lead to heart abnormalities, can lead to calcification of the heart. So there is a threshold for exercise in terms of its being beneficial. It's pretty rare. And we know of other things that obviously go wrong with exercise if you do too much, like amenorrhea for women, or bone fractures, again, things that I mentioned in the last podcast. So having too much exercise, which is difficult to do, but it's still possible, can have detrimental impact. So there's, it ramps up, and then eventually you hit kind of this point where you don't really get any benefit. And if you really try to just continue to push it, do more and more and more, that not only don't you get a benefit, you start getting decrements in your health. So that's they're kind of looking at this in a molecular sense or a cellular sense, not just a physiological sense or kind of a whole body physiological sense. But here they're just pointing out, hey, exercise. We know that exercise can be detrimental because it has been shown to be detrimental towards the heart, if, again, in excess. And finally, the last thing I wanted to quick mention before I go into the study design is that this mitochondrial dynamics. So the way that mitochondria behave is based off of two different processes called fusion and fission. And while physicists have their own levels, their own type of fusion and fission, cellular biologists or molecular biologists also have their type of fusion and fission, and they're not the same. So fusion and fission when it comes to mitochondria is fusion is two mitochondria coming together and literally fusing. So they open up and they fuse together to create one much larger mitochondrion. And fission is the exact opposite, right? So you have one mitochondrion and it pinches down the middle, or it pinches at a certain point, and then it separates out into two smaller mitochondria. So that's how mitochondria, one of the mechanisms by which mitochondria can change their shape and change their function and things of that nature. So that's where I'll leave that, and of course I'll be going more into that as we get deeper into this. So a bit on the study design. The researchers recruited 6 women and 5 men to train, and they were supervised. So they trained for 4 weeks using high-intensity interval training with increasing difficulty each week, so with more training each week. So if you're watching the podcast, you can see that I've got this graphic up, and it's really just showing the amount of volume, so the amount of work that they ended up doing. So they had a baseline where the researchers did measurements, and the measurements were a muscle biopsy. So they took muscle tissue from their legs, and then they also did an oral glucose tolerance test. We're not interested in that in this podcast. Again, refer back to the last podcast if you're interested in that. But we are interested in the muscle biopsies because we're going to get a lot of information out of that. So they did muscle biopsies at the baseline, so before they implemented this exercise protocol. And then the first week was a light training week. So they did 36 minutes of high-intensity interval training. The next week, so week 2, is moderate training. They did 90 minutes of high-intensity interval training. And then the third week, which was the excess training week, they did 152 minutes of high-intensity interval training. And then week 4 was a recovery week where they dropped back down to 53 minutes of total high-intensity interval training. And that's how it's broken down. And they did muscle biopsies throughout those 4 weeks. So the study's 4 weeks long, and we're getting measures of the cellular components. Okay, so now let's look at the data. So the first piece of data that I'm going to be talking about is fusion, mitochondrial fusion. So mitochondrial fusion is regulated by a variety of different proteins. So, right, you have your cell, and you have mitochondria that are within that cell. And the mitochondria have 2 different membranes. They have an outer membrane and they have an inner membrane. And that outer membrane has to essentially hook on to another mitochondria, or mitochondrion, because mitochondria is plural. Whatever, I get stuck in my own head sometimes. The point is that you have proteins that are on the outside, on the outer membrane of the mitochondrion, and they will hook on to other proteins of the other mitochondrion. And once those hook on, then they can start to fuse the membranes together, the outer membranes together. And then eventually you have the fusion of the inner membranes as they come together as well. And then you create this one much larger single mitochondrion. So the proteins that they decided to look at were proteins. One protein that is on the outer mitochondrial membrane. So if you were to just look at a mitochondria, if you were inside of a cell and you were just looking at a mitochondrion just kind of floating there, you would be able to see this protein poking out. It would be very visible. Now you would have to cut through that to see the second protein, but the first protein that is on the outer mitochondrial membrane is called mitofusin. Now, interestingly, they looked at mitofusin 2. Typically, you'd want to look at mitofusin 1, but maybe that's because it's specific to muscle. I don't know. But it is a fusion protein, so it is extremely important for the actual fusion of these mitochondria. So they looked at mitofusin, which is abbreviated as MFN, and that is, again, on the outside. And then on the inside, they have another protein called OPA. And these actually work together to end up creating the fusion of two different mitochondria. Okay, so what did they end up finding? By comparison to the excess training, so the excess going overboard with their training, they found that there was a kind of a stepwise increase in mitofusin and a stepwise increase in OPA1. So both of these fusion proteins increase as exercise volume, or the amount of exercise, increases. So that's really interesting. And that's going to kind of build into this story. Some of it is going to be a bit confusing, not because I'm trying to make it confusing, because the data is confusing. But that particular piece of information is pretty poignant. So then the next thing, obviously, to look at is, okay, fusion, good. We see that there's an increase in these fusion proteins, so maybe there's an increase in fusion. But now we're interested in fission, which is the exact opposite, right? The separation. And what they ended up showing is that DRP, so DRP is a protein that is found in the cytosol, so it's actually separate from the mitochondria. And what happens is it gets triggered to go to the mitochondrion, and then it interacts with a variety of different receptors. And these receptors are fission receptors, and they come, there's a bunch of them. There's a variety of different ones. So I'm only going to be talking about the ones, or a few of the ones that they mentioned, which are MFF and Phys1. And essentially DRP interacts with these, and then it essentially allows for the pinching of one large mitochondrion into two smaller mitochondria. So it'll usually pinch it down the middle, but essentially the point is that it pinches and continues to pinch, and it's a very cellular, energetically expensive process. And then you have the two separate mitochondria. So if you have DRP interacting with these DRP receptors, if that's MFF, if that's Phys1, if that's mid-49, mid-51, whatever it is, then you typically would think, okay, maybe there's greater fission then that's occurring as well. So I'm not showing the data for DRP, but DRP was actually not changed. So there's no change in the expression of DRP. However, there was a change in the expression of these mitofusin proteins. So not mitofusin proteins, in these DRP receptor proteins. So MFF and like Phys1. So with MFF, there was a decrease in its expression, in the amount of MFF with excessive training. However, interestingly enough, there was an increase relative, by comparison to the baseline. There was an increase in Phys1. So that's where things get a little confusing, right? So you're getting the decrease in one of the fission proteins, and you're getting an increase, or relative increase, in another one of the fission proteins. And yet the DRP, the kind of the main modulatory protein, does not change. Now, that raises some interesting questions. Maybe this is obviously saying that fusion proteins increase and fission proteins are kind of all over the place. So clearly we need more data, but whatever fission might be occurring is likely occurring through Phys1 over MFF, because MFF is starting to decrease, even though DRP levels are not changing. So that this ultimately leads to inconclusive results based on the data provided, except for the fusion that was relatively consistent. However, really what these researchers should have done is done some microscopy experiments, meaning that they should have taken images and actually visualized, seen these changes in the mitochondria as opposed to just looking at the proteins, because the proteins are giving you one story, sort of a story, half of a story, and then a really confusing other half of a story, like the buildup of a great movie for the first half, and then it just kind of like goes all over the place, and you're like, what? That made no sense. That's essentially what's happening here. So it would have been nice if they'd had some images to go along with this to actually show what is actually happening, and then maybe they can reconcile some of the differences. So, for example, you could see no change in DRP, right? No movement of DRP from inside the cell to. And the mitochondria are also inside the cell, but from another area of the cell to the mitochondria, maybe you don't see an increase in the actual amount of DRP, but maybe more of it will move to the mitochondria. You're missing that with this kind of experiment. You're only looking at the total amounts of DRP. So I think it's a pretty fair and understandable criticism, and I really wish that they had gone far more in depth with this, because ultimately the only thing we can kind of say we have a little bit of evidence for is that you have more mitochondrial fusion. And even that would require a lot more information. Okay, so however, then they also looked at mitochondrial antioxidant production, so the ability to buffer these reactive oxygen species. They also measured reactive oxygen species, so how much are the mitochondria creating these destructive molecules, and they then also measured this signaling pathway that I had mentioned earlier with KIAP and NRF. So to look at these antioxidants, so I'm only showing data for one, but they looked at a variety of different antioxidant proteins. And again, these are proteins that neutralize reactive oxygen species. And so they looked at ones like superoxide dismutase and glutathione, and I believe one other one, maybe catalase or something like that. But ultimately, the point is that they looked at a variety of different antioxidant proteins to see if the expression was increased or decreased or didn't change with this excessive training. And with the excessive training, there was no change. So I realized that if you're looking at the data itself, you might think, well, clearly it's a little bit higher. But statistically speaking, there was no change. So that's what we have to go off of. And all right, so maybe there's no change on that side, but maybe there's a change in reactive oxygen species, right? Production of, maybe you might think it's excessive. Maybe there's more of this reactive oxygen species production. Well, it turns out that is not the case. And I'll explain that in just a second, but really, where do these reactive oxygen species come from? If you haven't seen my content that I just released just a couple days ago when this actually goes live, I have a few videos explaining why we breathe oxygen and why oxygen is killing us. Those two videos explain where these reactive oxygen species are coming from, but essentially you have a series of proteins within the inner mitochondrial membrane, so inside the mitochondria, and they produce energy, and in the production of energy, they require oxygen. So they offload electrons or subatomic particles from the different proteins that make up what's called the electron transport chain. So these 5 proteins, or 4 or 5 proteins, use electrons, those are subatomic particles, to essentially charge up or allow for the production of cellular energy. I don't want to go too much into detail with that for this, because it's going to get really bogged down with a lot of stuff. But ultimately, when you have a mismatch in the amount of electrons that get passed on to this oxygen atom or molecule, then you get the production of the reactive oxygen species. So it's off of the production, and I mentioned this earlier, the production of cellular energy, that we get this production of reactive oxygen species. So what do we find? Well, with excess training, they actually showed a pretty dramatic decrease in the production of reactive oxygen species. This was measured at a variety of different places. So this is complex 1 and complex 2, so the first two proteins in the electron transport chain. I wish that they had measured it at complex 4 as well, but who knows? I don't know. Maybe they just they could only do 2 or 1, or whatever it might be. But ultimately, the results were actually consistent with a few different measures. I'm only showing one piece of data, but they ended up showing that there was a consistency in terms of a decrease in reactive oxygen species. Now, interestingly, you see an increase with the light training and the moderate training, and then a sudden sharp decrease with the excessive training. So clearly there's a difference there, and obviously the volume is significantly different between those. So then, and I'm not going to show the data on this, but then another thing that they were interested in was looking at lipid peroxidation and protein carbonylation. What does that mean? Well, lipid peroxidation is essentially when you have free radicals, or more specifically in this situation, reactive oxygen species that end up doing damage to fats that are within the cell. If that's part of a membrane, if that's part of a signaling molecule, whatever it is, ultimately it's doing damage to specifically fats. So they wanted to measure that, and then they measured the amount of damage that was occurring to the proteins within the cells, which is called a carbonylation. And they, by comparison, across all measures, there were no differences in the damage. Now, why might that be, right? So we didn't see any changes in antioxidants, but we did see a decrease in the reactive oxygen species. So maybe the level of reactive oxygen species production in the light training, and even at the basal level and even at the moderate level, that is a kind of a normal amount of reactive oxygen species, and that's actually being used as a signaling molecule. And the dampening that we see is maybe some sort of negative effect that's not actually causing more damage, but it's maybe reducing the signaling that's occurring through the reactive oxygen species. Who knows? Ultimately, that's you could come up with a few different hypotheses as to why that might be the case. Why you see a decrease in ROS, reactive oxygen species, compared to the other exercise conditions. Now, finally, the last thing was to look at this mechanism, right? So if we take the data from the previous podcast and talk about the data that we've discussed here, they were interested in, okay, well, one of the mechanisms for this is through this NRF KIAP system. So if you'd like to see the actual visual of this pathway, I've created a visual for it, but I'll describe it. Essentially how it works is that KIAP is a negative regulator of NRF. What does that mean? Well, KIAP, the protein, will bind NRF, and it will keep it in what's called the cytosol of the cell, so the main section of the cell. It'll keep it just sequestered there. And eventually what ends up happening is NRF can get what's called ubiquitinated, or really, in a more layman's terms, it gets tagged. And once it's tagged, it gets degraded by another protein called the proteasome. So the proteasome is responsible for taking tagged proteins, specifically by ubiquitination, and degrades them or breaks them up into single amino acids as well as double amino acids, triple amino acids, just smaller components, essentially just destroys the protein. So clearly, if you have a destruction of NRF, then it's not going to be able to go into the nucleus. So a movement from the cytosol to the nucleus is called a translocation. So it's moving from the cytosol to the nucleus. And once it gets there, what it would typically do is bind genes that are responsible, I mentioned this earlier, responsible for the production of more antioxidants, as well as, which are like the superoxide dismutase that I mentioned earlier, or and also the mitochondrial production, so production of more mitochondria. So that occurs. The distinction between those is through what's called a phosphorylation event. So if the NRF protein that is stuck to KEAP is phosphorylated, meaning that a phosphate group is added to it, then it will dissociate from KEAP. So it will separate itself from KEAP and be able to move into the mitochondria into the nucleus to affect its change that it needs to do, to bind its genes and allow for the production of all these proteins for antioxidants and for mitochondrial production, as well as other things as well, but that's what we're focused on. So what do we find? What do we actually find with the exercise then component? Well, the NRF actually decreases with the excess training. So there does seem to be, although I don't think that there's a statistical significant increase, but it seems to me like there's an increase in NRF protein when it comes to the light training and moderate training. However, with the excess training, it then has a stark decrease. Maybe not by comparison to the baseline levels, but definitely by comparison to the other exercise conditions. And then what you would expect is that you would see an increase in KEAP, and that's actually exactly what we find, that KEAP levels increase and NRF levels decrease. So what's presumably happening here is that you have more KEAP that's binding the NRF in the cytosol and then leading that to be ubiquitinated, and then it gets shuttled to the proteasome, and the proteasome then degrades NRF. So this sort of makes sense, and it sort of doesn't make sense. But let me kind of give you the takeaways from here. So excessive exercise shows decreases in the reactive oxygen species production that we talked about, but also no increase in the antioxidants. So NRF2 is reduced and KEAP is elevated. So that implies then that there's a reduced production of antioxidants in mitochondrial biogenesis, or the production of more mitochondria. We don't have a whole lot of great evidence for the mitochondrial production, but we did look at antioxidants, and we saw that there was no effect. So, maybe the antioxidants are being stabilized somehow. It's possible that the ones that are already produced, even though you're producing less, maybe you're not degrading them as quickly. That's a possibility. Or there are other kind of bigger possibilities, which I'll actually go ahead and go into right now. So the conclusions, the takeaways from all of this information. Excessive exercise leads to increased markers for mitochondrial fusion. So, the combination of two mitochondria into one, larger one, but inconsistent results for mitofission. There is no increase in free radicals, so those reactive oxygen species, nor is there any sort of effect on damage to the internals of the cells, that lipid peroxidation and the protein carbonylation that I was talking about earlier, and actually leads to a reduction in reactive oxygen species production. This may be due to reduced levels of NRF2, which is a protein that increases mitochondrial production as well as antioxidant production. However, it may be, and this is what I was alluding to earlier, that it may be that long-standing effects, negative or positive, are still unknown. So they're doing these muscle biopsies, I believe, 14 hours. I believe it's 14 hours. I could be off on that, but it's within a day of their exercise training. So if they do it maybe an hour afterwards, or if they do it maybe a week later or 2 weeks later, or whatever it might be, could you see different effects? Most likely, yes. And maybe you might see some downstream things, because 1 week of excessive exercise may not be long enough, or the time points of muscle collection were too soon, which is what I just mentioned. So, but on top of that, it's possible that muscle cells prioritize combining the existing mitochondria instead of production of new mitochondria. That, I think, is pretty plausible. If that's a negative effect, however, is unknown because we don't actually know how that ends up panning out long term. It's unknown, and it actually may not be. It may just be a difference. It might just be a different way that the cells are reacting to excessive exercise. So instead of producing more mitochondria, they're just maybe making mitochondria more efficient, or there's a number of reasons why mitochondria might undergo fusion. So this study is broad and it's definitely far-reaching. So I have critiques, I have criticisms of this study, although it's a great first step. It's a great kind of first dipping your toe in the water. But it covers so many different things. In the last podcast, it covered some aspects of metabolism. We talked about glucose tolerance, so blood sugar tolerance, insulin sensitivity. I mean, a bunch of different things. And then here they're talking about reactive oxygen species, and they're talking about signaling pathways, and they're talking about mitochondrial dynamics. So there's just a lot here that they've kind of packed in, and it feels like they've just kind of lightly peppered over everything. They haven't been like, oh, this is interesting, therefore we'll dive into this particular point. So it's broad and far-reaching, but the conclusions from this podcast and the previous are a bit premature. So it's kind of hard to make really definitive conclusions. Some interesting data for sure, but hard to say, oh, excessive exercise, definitely bad in every way. It just might be different. Not necessarily bad, but different, which is also possible. So the data simply doesn't dig deep enough, which is what I just mentioned, to assume excessive exercise is immediately leading to impaired function of mitochondria or metabolism. So more data is absolutely needed with more measures of these processes. For example, with the images, they should have taken images. That's actually pretty much like standard. You have to take images if you're going to be doing these different fusion and fission assays, and also at different time points. That would have been really nice. Okay, folks, hopefully you found this informative, and if you didn't check out the last podcast, I certainly recommend it. And with that, thanks for stopping by as always, and I hope to have the absolute pleasure of speaking with you in the next one. See ya.